So thanks for joining us today on this webinar about the GEOTRACES program. My name is Elena Masferrer. I am the Executive Officer of the GEOTRACES International Project Office, and I will introduce you this event on behalf of GEOTRACES. During the talks, you will be mute and your video, please, will be off. You can use the chat at any time to submit any question. The presentation will be recorded and will be available on the GEOTRACES website as well as the Authenticate website. Please help us to give publicity to this event by posting about it on the social media. Next slide, please. Thank you. The program for today is shown in this slide. First, you will watch a 2-minute video about the GEOTRACES program. Then Maite Maldonado from the University of British Columbia in Canada, who is also co-chair of the GEOTRACES Data Management Committee, will explain you about the GEOTRACES program and the lessons we learned while creating the GEOTRACES product. Professor Rainer Schlitzer from the Alfred Wegener Institute in Germany and senior developer of the GEOTRACES product and Web of the Week, which is a web tool for extraction, analysis, exploration, and visualization of oceanographic data, including the GEOTRACES data, will introduce the GEOTRACES data product and show how you can access to it. Finally, Maite will give an example of potential uses of the GEOTRACES data. Maite will also moderate the debate at the end of the event. Thank you very much. So we will start now with visualization of a video. I ask, please, Chris, to display it. Thank you.
Let's imagine that we are able to see our future in the oceans. Does this idea seem surprising to you? And yet, marine environment shelters a large amount of information which, once decrypted, gives us the magnitude of mankind's impact on the ecosystems. So it's possible to anticipate the amplitude of global warming and prepare tomorrow's world. This is the mission the GEOTRACES program contributes to since 2010, bringing together scientists from 35 countries. Given the sheer size of the oceans, they had to unite their efforts to develop technical solutions essential for detection of chemical elements in the seawater. These elements include nutrients essential for marine life, such as iron, zinc, manganese, and copper. Toxins that accumulate along the food chain, such as mercury and lead, or radioactive elements. GEOTRACES also measures the indicators that provide information for improving climate models, such as beryllium, radium, thorium, and rare earth elements. As an example of the complexity of this process, quantifying the iron in a sample is like measuring the amount of metal present in a paperclip dissolved in 30 Olympic swimming pools. To this end, oceanographers in the GEOTRACES program are organizing major scientific cruises to explore the widest reaches of the world's oceans. These expeditions can last more than a month, during which experts carefully collect samples using clean techniques and specialized equipment throughout the depths of the ocean. The extracted samples are then transported to home laboratories before being analyzed using results which are assessed and shared. This valuable data will complete a global atlas showing the distribution of multiple trace elements throughout the ocean. Now everyone can benefit from this freely available information and use it to answer questions about our changing world. As you can see, GEOTRACES' mission is a major challenge in line with the current environmental goals, in which we can all take part and change the world together.
So before I start, I would like to bring your attention to the 10 frames behind me in this slide. And in essence, these are 3D images of various ocean basins with the distribution of some key elements, which you can find in the eGEOTRACES Electronic Atlas, which is part of the latest GEOTRACES Intermediate Data Product, or what we call IDP2021. So what I would like to do today is to share with you some of the lessons we have learned while creating this product. Next, please. Next slide, please. For those of you who are not familiar with GEOTRACES, GEOTRACES is an international study of the chemistry of trace elements in the ocean. GEOTRACES' mission is to identify processes that control the distribution of key trace elements and isotopes, and to establish the sensitivity of these distributions to changing environmental conditions. The ambition of GEOTRACES was to map the global distribution of these trace elements and isotopes, and this undertaking is only possible by the collaboration and coordination of multiple nations. Thus, scientists from over 35 countries are involved in GEOTRACES, which is designed to study all major ocean basins. The scientific plan was finalized in 2006 after a series of international workshops where countries came to the table with their own research questions of interest, their infrastructure, and their limitations. The idea was not to prescribe a program, but to design a program that will allow many nations to contribute in many different ways. For example, a nation could contribute a major cruise, or they might contribute the creation of a data management center. The first cruise took place during the International Polar Year in 2009 and 2010. And one of the key decisions of this program was to release intermediate data product throughout the life of the program, instead of just one product at the end of the program. We have had 3 intermediate data products, and the last one was released in the fall of 2021. Next, please. Trace elements are elements in the periodic table that are present at extremely low concentrations, but play a vital role in controlling the biology of the ocean, both as essential nutrients or as contaminants. GEOTRACES aim to map and understand the processes that control the global distribution of trace elements and isotopes in the ocean. The data allow us to gain a better understanding of, for example, how trace elements control oceanic productivity and ecosystem functioning, or the fate and impact of contaminants. We can also learn about how climate change is affecting physical, chemical, and biological processes in the ocean. We can also use various isotopes to investigate past climate or changes in the circulation of the ocean that are occurring right now. Some specific examples I would like to share with you that we have learned from the GEOTRACES data are the following 3. Next, please. With GEOTRACES data, we have been able to document some of the climate-mediated changes that are happening in the Arctic. Radioisotope 228 is an excellent tracer of land-ocean inputs. Measurements of radium-228 in 2015 by the US team in the Arctic revealed that the concentration of this isotope in surface waters had almost doubled in the last decade. A mass balance model allowed Kip and colleagues to suggest that the increase of radium-228 in the surface water was mediated by intensification of shelf-derived inputs from marine sediments. This was in turn due to the defrosting of marine sediments. These coastal changes could deliver more nutrient, more carbon, and other chemicals into the Arctic Ocean, and could lead to dramatic changes to Arctic food web and marine mammals. Next, please. GEOTRACES data can also provide evidence of the efficacy of major legislations to reduce contaminant emissions. We are really concerned, for example, with mercury and lead. Human activities such as the combustion of leaded petrol or emissions from non-ferrous metal smeltings, coal combustion of waste incineration, all contribute to the release of lead during the past century. This has resulted in a considerable increase in anthropogenic lead in the North Atlantic. And it's been large enough that actually masks the natural lead signal in these surface waters. The phasing out of the leaded petrol since the mid-1970s has decreased this contamination. By measuring lead concentrations and isotopes along 2 GEOTRACES cruises in the North Atlantic, Bridgestock and his colleagues were able to measure again for the first time the natural lead signal in surface waters of the North Atlantic. And they were able to identify the source of this natural lead to Saharan mineral dust. These data clearly reflect the success of the global efforts to reduce anthropogenic lead emissions. Next, please. GEOTRACES data also can be used as a baseline to assess future anthropogenic inputs into the ocean. What I'm going to present to you here is an example using rare earth elements. Rare earth elements are starting to be used by various industries now. And for example, gadolinium is now widely used as a contrasting agent in magnetic resonance imaging, leading to its release in the environment. Although we are not yet able to detect gadolinium in the open ocean, we can actually measure gadolinium in coastal waters. And Pedreira et al. Suggested that we can use gadolinium as a tracer of wastewater into coastal waters. GEOTRACES data may thus serve to assess future changes in the natural distribution of rare earth elements and provide unique tracers for anthropogenic inputs from various industries. Next, please. GEOTRACES scientists needed to determine the global distribution of many elements that are extremely at low concentrations in the ocean and are also often prone to major contamination. To achieve the goals of the program, the GEOTRACES community needed to develop, implement, and share contamination-free methods, as well as expertise and infrastructure. Capacity building was essential to achieve the goals of GEOTRACES. Next, please. To give you an idea of the capacity building by the GEOTRACES program, we have gone from having 4 laboratories making accurate measurements of iron in the ocean to over 20, including laboratories in China and India and so on. This has been a real joy to see and experience. GEOTRACES offer international assistance in designing, constructing, and using clean sampling systems. It also offers expertise and training through summer schools and workshops. And also there is a wealth of online resources like best practices and tutorials, etc., that you can find in the website. One of my favorite is the GEOTRACES cookbook, which is a wonderful manual with detailed explanations of how to sample for all the elements that GEOTRACES is measuring. And it's used in my laboratory almost every day. Next, please. So some of the important aspects that have made the GEOTRACES data product possible are in this slide. The first thing is, in order to accomplish this, we really needed to have an incredible international collaboration and coordination. The science plan in 2006 was key for the coordination of such an ambitious global research program involving multiple nations. The science plan allowed for the optimization of resources and ensure adequate ocean coverage with minimal unnecessary redundancies. Under the umbrella of Scientific Committee on Oceanic Research, GEOTRACES coordinates the research activities of more than 35 countries, and national representatives are key as they organize the research activities and scientists in their own country and act as liaisons between individual countries and the GEOTRACES Scientific Steering Committee. The GEOTRACES Scientific Steering Committee oversees the program development and coordinates the research activities of all the countries. This coordination is ensured, in turn, by the International Project Office in Toulouse, led by Catherine Jendal and Elena McFerrer. In order to build a common ocean atlas, the quality and the internal consistency of the data are rigorously controlled by intercalibration protocols and supervised by the Standard and Intercalibration Committee. Once the data is validated and under the supervision of the Data Management Committee, the data are sent to the GEOTRACES Data Center at the British Oceanographic Data Center in Liverpool, where the data is processed and finally is made available to the public in an international open access database. Next. To give you a summary of what we have accomplished, as I said, the GEOTRACES cruises started in the International Polar Year. These sections are marked in the map here in black. The Arctic Ocean was fully completed in the summer of 2016. The sections that you see here in yellow are those that we have completed since 2010. You can see that most of the Atlantic Ocean has been completed now. The sections in red are those that are being completed right now or are in the planning process. We still need to cover a lot of the Pacific Ocean as well as the Southern Ocean, which is extremely difficult to explore due to the rough marine conditions and its remoteness. GEOTRACES cruises must follow defined criteria and be approved by the GEOTRACES Scientific Steering Committee. Next, please. We decided to set up a committee dedicated to the quality of the data because we are a large international program with many nations. The production of trace metal and isotope data by multiple labs in each of several nations, often employing novel methods for challenging measurements, require rigorous quality control to ensure internal consistency throughout the global suite of geochemical data. The standardization and intercalibration of the data results in high quality data across all stations in the ocean, allowing us to generate a world map of the distribution of key elements and isotopes. This committee is responsible for sharing resources, expertise, and equipment when needed. It's able to recommend sampling protocols. This is the cookbook I was telling you about. Also, it's going to give advice on how to intercalibrate your data. More on this in the next slide. And also is able to distribute reference seawater samples to various analysts. The Standard and Intercalibration Committee evaluates all the data that was included in the intermediate data product to ensure accuracy and assist investigators who experience data quality problems. Next, please. Intercalibration is an ongoing process in GEOTRACES. Each lab is expected to demonstrate intercalibration of their data for each cruise. There is a constant dialogue between this committee and the analyst in order to obtain coherent and high quality data. They are not there to judge you, but to make sure and to help you get the best high quality data possible. For trace metals for which reference seawater samples are available, these reference materials are provided to the analysts as working standards to assess the performance of their method and the quality of the data. For trace elements and isotopes with no reference materials, there are several options for intercalibration. One of them is so-called crossover stations intercalibration, which involves measuring the element of interest at multiple depths at a given oceanic location by two different countries. These data are submitted to the Standard and Intercalibration Committee by each country. The committee examines the quality of this data. The data is then submitted, the data normally submitted by each country, normally match beautifully at depth away from the surface processes. What is unique, I think, about GeoTraces is that there are multiple ways to intercalibrate the data. And this flexibility allows incorporation of high quality data from many countries. And the multiple ways to intercalibrate your data are indicated in those middle arrows in this slide. Orange, green, purple, all those arrows are all the ways you can intercalibrate your data. Next, please. Data management is a process that is a process that every big program such as GeoTraces needs to perform. The main purpose of the data management is to combine individual data sets produced by the different international laboratories in a common framework in one big and coherent high-quality data set. That means bringing together data from different countries and from different groups, measured at different times and locations, but also bringing together data for different parameters. So in essence, this is an integrative process that ultimately allows the creation of a big, harmonized global data set. The Data Management Committee provides oversight and interfaces with data generators. For example, they recommend data policies, standards, and format for data submission. After the creation of the intermediate data product in 2017, it became obvious to us that we needed to have a better way to register the data sets. So we created the DOOR portal. The Data Management Committee is in close contact with the Data Assembly Center, who is responsible for compiling the data in an organized and accessible structure. This committee is also responsible for formalizing parameter names and for creating fair use agreements for data access to all. Next, please. This is a picture of the data portal, the website of the data portal, DOOR, that I was mentioning. I would like to thank Elena Masferrer, Francois André, Julian Brissett-Brad, and Arnaud Muir for the creation of this portal. This has been essential for this last data product. This DOOR portal was designed to streamline the preparation of the IDP2021, both for the principal investigators and those who work behind the scenes to assemble the data product. The purpose of the DOOR portal is to facilitate the registration of the data and the tracking of the data, as well as the submission of the intercalibration report for processing by the Standard and Intercalibration Committee. Next, please. In the DOOR portal, you can quickly and easily register your data and your metadata. It only takes 15 minutes to do so, and this is because there is all these pre-filled forms and templates. All you need to register your data is an ORCID number. You can then track your data very easily because as soon as you register your data, you get a barcode for your data. There are 6 interfaces in this data portal for different users and people behind the scenes. There is one for the principal investigator to register the data and track the status of the data. There is also an interface for the Intercalibration and Standard Committee. There is also an interface for the Data Management Committee. Also, the Parameter Naming Committee has their own interface. And the International Program Officer also has an interface where Elena, for example, can link data sets to publications and so on. The principal investigators can use the DOOR at any time to follow the progress of the intercalibration report, to link publications to their data, to associate scientists to the data, or to give permission for their data to be released in the next data product. This portal is also wonderful because all the data is centralized and the metadata. So this allows us to go to the data product and click one little button and download the entire data set that is of interest to you. So with that, I would like to introduce you to Rainer. Next slide, please. Rainer Chitzler is the senior developer of WebODB and the data product, and he's going to tell us about how to access this data product and all the cool things you can do with it. All yours, Rainer.
Thanks, Maite. Hello, everyone. I'm Rainer Schlitzer from the Alfred Wegener Institute in Germany. In the next slides, what I want to do is show you how you can access and use the data from the IDP 2021. Next slide, please. GEOTRACES as a program is very keen to maximize the usage of its data. The program is proud of its data. They are excellent, new, high-quality data, and the program wants them to be utilized in the widest possible way. So what I want to show is how easy and flexible and the different ways for you to access these data. So the first thing we need to know what's inside. Actually, the IDP 2021 consists of two pieces. One is the actual numerical data. And second, there will be, or there is the electronic atlas. So it's a gallery of visuals that are pre-created for you to look at and to study, the eGEOTRACES Atlas. And we are going to see how to access both in a minute. So next slide, please. A natural way or entry for getting access to these two components is through the GEOTRACES website. www.geotraces.org. You go to the data tab, and on the left side, the list on the left side that provides you with the links. And we will start with eGEOTRACES. Next, please. So can you go back? Yes, so in the middle, we have the eGEOTRACES Atlas link. So you click on this link and then next slide. So that will take you to the eGEOTRACES Atlas. That's a website maintained at Alfred Wegener Institute. eGEOTRACES.org is the address. When you go there, you see a map like on the left side here in the slide. You choose a tracer group in the element on the left top side. Once you, like here in this example, we have chosen dissolved trace elements. In the item to the right, you choose one of these elements. So in this case, dissolved iron. Once you have chosen a group and then the TEI inside that group, in the map, all the sections that have data for this particular choice you have made, they turn red, and the labels of these sections, once you click on one of these labels, like here in this example, in the South Pacific, the label indicated by green here, once you click on it, then you will see the section for this particular tracer you have chosen along this particular line, like in the window here to the right. Not only do you see the graphics of these tracers along the line, but in the lower part of the page, there's important information. One is a description of this element. For dissolved iron, this is kind of easy, but be assured, GEOTRACES has complicated TEIs, and you appreciate having a description of the TEI here. Below that, you see the list of scientists who have created the data. We think this is very important. Making these measurements is so delicate. We want to honor the people who do this work. So all these names are here. And then you have groups of items that take you further. You can either choose, you stay along this same section, but if you want to see a different parameter, you can click on one of these items. If you want to stay with this tracer, but you want to see it on a different line, then you can easily click one of the other GEOTRACES lines. Or if you want to see a 3D scene, then you choose the ocean basin and click on that. And you would get a 3D animated scene like shown in the lower right part of the slide. eGEOTRACES is a vast resource. I invite you to use it heavily for scientific purposes, but also for teaching and making presentation. Overall, there are almost 1,500 sections and more than 260 3D scenes. So there's a lot of things you can look at. Next slide, please. So when you want to get at the numbers themselves, at the actual data themselves, we have set up 3 ways of accessing them. And the links to these 3 ways of getting to the data, they are in this orange box here in this slide. And we will go through them one by one. Next slide, please. So number 1, if you are a person and you want to have everything in a package and you want to carry around everything, maybe you are disconnected from the internet and you just want to make sure you don't lose anything, then the first way of data access, that's for you. We call it bulk download, basically meaning you will get the full package. Very often, you don't want the full package. Maybe you are focusing on a specific domain. Maybe you are focusing on a specific range of parameters. For those people, we have set up an online service that lets you subset. So you can filter out what you want and then extract only this part of the data and download this one to your computer. And the third way is you don't download anything, you do the analysis and the visualization of the IDP data truly online. And that's number 3, and we will get at that also. One comment I want to make is, with previous IDPs, we required that people register and then log in with their credentials before they get access to the data. This has changed with IDP2021. You will be shown what we call the fair data use statement. You have to agree to these rules. Once you do, you get access. You can stay fully anonymous, no registration necessary. Next slide, please. So let's do bulk download. We download everything. The link highlighted here on the slide. Next. So that will take you to a site at the British Oceanographic Data Centre. The web page is shown on the right here. In the middle, there's a download button that actually lets you download the files. And what you will get is all the individual data sets that we have, consisting of sensor data, the actual core data of GEOTRACES, seawater discrete sample data, you get the sensor data, you get aerosol data from the atmosphere, you get precipitation data, and a rather small data set right now, some cryosphere data from snow and ice. And you get all of them, and you get them in these three formats: ASCII spreadsheet, NetCDF for modelers, and ODV collection for the large group of ODV users. So that's bulk download. You are set if you have it, and you can go to remote areas, even disconnected to the internet. Next slide. Now, if you want to subset and extract only parts of the data, then you follow the link highlighted here. Next. And that takes you to an online service that we at Alfred Wegener Institute are maintaining. The address is here, geotraces.webodv.awi.de. The subsetting and extraction service is done in a sequence of simple steps that are highlighted in the navigation bar near the top. First one will be selection of domain, time range, and cruises. That is shown here. In this particular case, we have focused on the South Atlantic and have just selected 3 cruises out of the many cruises of the IDP. The second step, next slide. Here you select the parameters that you actually want the data for, and you are presented in the middle lower part here, the tree of IDP TEIs. So you have the groups, and once you open the group, then you see all the individual TEIs. You can select parameters individually, but also you can select or unselect entire groups by clicking on the group box. Next slide. Third step, you choose a format, left upper part of the slide here. You click on one of these 3 formats, and in a matter of seconds, you will be presented with a zip file containing the data that you have selected in that particular format, and you have it on your machine. So that's a simple, easy subsetting and extraction. And then you have the part of the data that really interests you, and you work with that. Next slide. So for those who simply don't want to download anything or install any kind of software for the analysis, we have a third service, which we call the Data Exploration Online inside the WebODV Explorer. To reach that service, you follow the link highlighted here. Next. And actually, we are providing this analysis and visualization service at 2 addresses, which are highlighted here on the top. I will show you the Explore version, the second link. That takes you to a page like this that gives you access not only to oceanographic or GEOTRACES data, but also to environmental data from the atmosphere, ice, sediment, or rivers. The ocean part is the biggest. Next, please. To drill down to the GEOTRACES data, you choose Tracer, then you choose GEOTRACES, then IDP2021. You will see all the individual data sets that are available inside IDP2021. In this case, we want to look at the seawater data. Once you click at the GEOTRACES IDP seawater discrete sample data link, then next slide. You will actually enter a live data session inside your web browser. So what this slide is actually showing is the web browser canvas, and deliberately what we call the WebODV interface that is mimicking the ODV standalone interface, and many people are using the ODV. If you do, then operating the web version should be easy. Just like in the standalone ODV, it's interactive. You left click to select samples or stations. You see the values of the metadata or the actual data in these lists on the right. If you right click on elements here, you get menus just like in the normal ODV, and the web version gives you functionality almost identical to the ODV itself. So in this case here, this would be a live session of ION along GP19 that we have seen statically in the eGEOTRACES several minutes ago. So in summary, it's ODV-like interface. All eGEOTRACES sections have prepared views, so you can activate them quite easily by loading that particular view. And as I said before, there's no need for any data download or software installation. You just start working. So this is service number 3. Next slide. So in summary, what I want to emphasize, IDP2021 really is an exciting new resource, and it has lots of rich information in it. We invite you to use it for research, teaching, outreach activities, and also to spread the word to people who might be interested. Next slide. I want to emphasize at the end that creating this IDP 2021 was a massive enterprise. It's a real group effort. A large number of groups and a large number of individuals have contributed. Without their work, this would not have been possible. So thanks to all who made it, and also what should never be forgotten, thanks to all the people who created the data in the first place. So thanks to the entire group. And this is the end of my presentation. And next slide. And I think I'm handing over now again to Maite, who will continue. Thank you.
Thank you, Rainer. So what I would like to show you now is how we try to link genomic data to the trace element and isotope data in the IDP 2021. Next, please. The GeoTraces community really wanted to strengthen collaborations between the marine geochemists and biologists and scientists that work with omic data. I have to thank many geochemists that were willing to collect samples for the omics lab in a series of cruises. And those cruises are highlighted in this map. Those gray dots that you see are stations where geochemists very nicely collected samples for omics labs. So you can see we have omics samples for a series of sections in the Atlantic Ocean as well as in the Pacific. Next slide. There are 4 types of omic data available in the IDP 2021. We have 500 metagenomes, 14 single-cell genomes, almost 300 rRNA gene analysis, 16S, 18S, and almost 500 functional and taxonomic analysis of metagenomes. Next, please. But we face a major challenge while we were trying to incorporate omic data into the IDP 2021. This is because the omic data is extremely dynamic and actually changes depending on the ever-growing genome reference databases, while the trace metal and isotope data is completely static. So a bunch of us got together. I would like to thank those people right now. So Rainier, Schitzer, Paul Berube, and Jesse McNichol, we had multiple meetings to figure out what's the best way to incorporate the data in the IDP. And at the end, what we decided is that the data could not be incorporated into the IDP, but we could provide data linkages within the IDP product. So what you are going to see in a minute is that we actually give you data linkages to the National Center for Biotechnology Information Database in the US, and we also give you linkages to the EMBL within the European Bioinformatics Institute database. I also would like to thank here everyone that have provided omic data, because this is super exciting, we think, in this data product. Next, please. So how do you find this data? These data were incorporated in the IDP 2021 as metadata variables. So if you go to the web ODB Explorer, you are going to find in the metadata 4 variables, number 11, 12, 13, and 14. This is where you can find, for example, for variable number 11, you can find the accession number for the metagenome data in the NCBI associated with that water sample. Similar for the variable number 12 and 13. For number 14, actually, what we have in the IDP is a clickable link that is going to take you straight to the website where the analysis have been performed already. So the way you link the omic data and the trace metal data is through the specific bottle number. This is very important. This is really what's linking the two data sets. Next, please. So this is another view of the Web ODB Explorer that Rainier was just showing us. So what I have done here, I selected as the variable of interest omic, the 4 variables that you see there, 11, 12, 13, and 14 that have a yellowish sort of highlighted. And so a map popped up telling me all the places where I have data for these 4 variables. So you can see those are all the cruces I told you at the beginning. So what you have here is 4 sample descriptions in the metadata variables. So you get accession numbers to the NCBI website, or you get a direct link to the EMBL website. So I want you to look at variable number 11 there, highlighted in orange, and you're going to see that it says NCBI metagenome biosample accession, and then you can read in capital letters SAM N07, blah, blah, blah. Next, please. So you take that number, that accession number, and you go to the NCBI website, and you just put it in the search bottom. So that's what I did right there. And you click enter. Next, please. And it's going to show up the metagenome associated with that accession number. And here you can see when the sample was collected, the depth, the station, and so on. And you, for those who are omics folks, you know what to do with this website. So you will do similar thing for variable 12 and 13. You will just get that accession number and go to the NCBI website and look for the data there. Variable number 14 is a little bit different. Can you go back 2 slides, please? Yeah, so if you go to the orange highlighter there, number 14, you're going to see that there is actually a clickable link. Move forward 2 slides, please. So if you click one more, if you click on that link, it's going to get you here. And this is the A EMBL database where actually the metagenome data has been analyzed for taxonomic analysis and functional analysis. So this is what you're going to see, and then you can click on those tabs that read taxonomic analysis or functional analysis. Next, please. So I click on the taxonomic analysis, and I was brought to here, where you can look at the taxonomic analysis of the metagenome using different illustrations, and I chosen the pie that you see here, and you can actually click in there and you can zoom in the taxonomic analysis. So you can really learn about all the organisms that are present in that water sample. You can also click on the functional analysis tab. Next, please. And if you do that, you are going to get the function of the genes that they were sequenced from that water sample. Next, please. Next. So I hope we have convinced you that GeoTraces data can really provide some wonderful clues about how to tackle, understand, and predict the future of the ocean and the planet. The data was generated following a global ocean science plan through international collaboration and coordination for sample collection across all ocean basins and data processing and management. We ended up with a data set that is very high quality for marine geochemical data. There are almost 2,000 publications associated with this data. The data is open access in a timely manner. That's why we released 3 intermediate data products so far. Not only you can find the data, but you can find those beautiful visuals within the data product that can be easily accessed by any stakeholder, educator, or the public. It's a wonderful resource. And I also want to emphasize that GeoTraces program provides and shares infrastructure, knowledge, and technologies for capacity building worldwide, which we believe is essential to deal with all the challenges that the ocean is facing today and is expected to face in the future. So next slide, please. So with that, I would like to thank everybody for coming. And if you have any questions for us, you can contact GeoTraces, the International Program Officer, or anybody else. And thank you very much. Subscribe to the e-newsletter, and we are happy to take any questions.
There is a question from Jim Bishop.
Jim, go ahead. Do you want me to read it? No.
No, actually, I don't have a question. I just want to thank everyone. I really enjoy the fact that data from GeoTraces is open, and I think both you and Reiner deserve a lot of thanks on that. So I put up my thumb.
Thank you, Jim.
Kevin Lindstrom has a question on the chat.
All right. So Kevin.
How are you tracking the publications? So actually, as explained by Maite, people is able to put the publication on the GeoTraces door. And as the door is designed, it's automatically displayed on a website that shows all the publications for each data point. Actually, from the WebODB and also from eGEOTRACES Atlas, people can click on a link and arrives to a website of any publication on this data point is shown. And this is done thanks to the help of the GEOTRACES door. Before I was doing that myself manually, but since the door has been there, then we can easily, our researchers can easily update the publications at any time, just going register in door and informing about this publication directly.
There's another question from Kevin, Elena. Our research.
Research is sitting BioDC dataset DOIs. Yes, they are. When actually in the fair use agreement, people engage to cite publication using the data.
Rainer Schlitzer speaking. I want to emphasize again that this ability to link the publications right next to the data, I would see as a strength of the GEOTRACES data system. So users will never have an excuse saying, oh, I used these data, but I didn't know there was a publication, because it's so easy in the IDP for any individual data point actually to find if there is a paper by the original researchers. And I really want to emphasize this is a strength, and it's thanks to the IPO and thanks to Elena and the developers of DOOR.
Yeah, and I guess I also would like to emphasize that in the fair use statement, when you read it, you will see that you are encouraged, if you are downloading data to do some modeling or whatever, you're actually encouraged to contact the data generators and maybe collaborate with them in the writing of the paper. So we really want to also enhance collaborations between modelers, for example, and data generators.
So I think we can conclude then, and I want to thank Rainer Schlitzer and Maite Maldonado for making these two talks, and also to Chris Parson, because we cannot see him, but he has been working, changing the slides and have been supporting all this webinar, and also to Stacy Aguilera-Peterson. Chris and Stacy are both from National Science Foundation, and I wanted to thank for all the help organizing this webinar. Thank you, everybody, for being there, and I'll see you next time.